did research with Dr. Bell this summer, and I'm continuing on through this year. And the name of my project is the effect of glutamine repeat insertions on the structure and function of GMDH. And Dr. Bell kind of already introduced GMDH, so I won't go too much into that. But to give you an overall kind of overarching picture of what we were kind of looking at here, we were looking into polyglutamine expansion diseases. They're a class of neurodegenerative diseases, and they're characterized by protein misfolding, which causes aggregation, and the aggregation causes the pathogenic effects in the diseases. So our main goals were to examine the effect of the glutamine repeat insertions on the structure and the function of malate dehydrogenase. Specifically, it's watermelon malate dehydrogenase, glycosomal, hence the GMDH. And we also wanted to look at or examine the structure of the actual glutamine insert in the protein if we were able to crystallize it. And GMDH is a ubiquitous protein found in all organisms, or most organisms, and it has a known structure and crystallization conditions. But the mechanism through which it folds is unknown. So we were hoping that by studying the way these glutamine inserts affected the structure, we would be able to kind of elucidate the folding mechanism of the native GMDH. And then we also were looking into the how the inserts affected the GMDH activity. Okay, so for our specific aims, we wanted to first start out by actually creating the mutant. So we were actually able to create 2 mutants. You can see here, let me see. Okay, so we were able to create 2 mutants, 240Q10, which is the 240 residue. This is the GMDH dimer. These are identical homodimer structures. So just over here, we have the 240Q10 mutant and the 301Q10 mutant. These are the 2 that we are actually able to grow up and purify, whereas the 301Q10 we weren't able to. So we kind of worked with these 2 mostly to get our results. And basically what we did was we inserted 10 glutamines at each of these sites. Okay, and so then we took the purified protein that we had, and we used MALDI-TOF, which is what Dr. Bell already kind of explained to you, to verify that we had the correct and expected mass change in the protein. And then after that we looked at the kinetic properties of the mutants, and then we initiated a structure determination. Okay, so these are our MALDI results. I only have 5 minutes, so I won't go into too much detail. But basically, you can see this is the mutant 301Q10 results, and this is the 240Q10 results. For these, you can kind of see here, this is the peak that we were really looking for, all 3 of these really, but this is the main one. And it shows that there's a correct molecular weight change that we would expect to see with the insertion of 10 glutamines in the protein. And then here, we also see the right peak in the 240Q10 mutant. However, we see a lot of other peaks. And so what we're thinking is that this protein, when 10 glutamines are inserted into it, it's misfolding and then somehow associating with other proteins, and when we purify it, it's being brought in with the GMDH. So that kind of goes back to what I was talking about with the neurodegeneration and the pathogenic characteristic of the poly-Q expansion diseases in terms of aggregation. Okay, so since we had more of the 301Q10 mutant, we were able to get enough of it to try and crystallize it. And after a bunch of preliminary studies of the crystallization conditions, we found one that actually worked. And so this is right there, you can see there and a little bit there. That's actually the crystallized GMDH. It's the mutated version. So here we used that crystal, and we were able to run X-ray crystallography using it. And we found that this is the data that we got from it. I won't go into too much detail, but see all of these little points. It's caused by the scattering of when the laser hits the crystal. And so we found that the GMDH301Q10 mutant, it follows the same space group as the native GMDH. However, it's in a smaller unit cell. So the structure is different. The structure as a whole has been solved, except we are still unsure as to how the glutamine inserts are actually forming. It seems to be kind of waving around and flapping around, so we're not sure. We haven't determined that exactly, but that's kind of where our efforts are being focused right now.